All right, so welcome to Module 6. This week we're going to be looking at DNA. It's the molecule of life, and we're going to be looking at it in a lot more detail. So last week we learned about chromosomes, and now we're going to learn about DNA and how DNA forms the basis of who we are. So the human genome in its entirety was not sequenced until 2003. So in the 90s, the Human Genome Project started, and it was a worldwide collaboration to sequence the human genome. And this was huge. When it came out that they had the first published human genome, they had sequenced it. It was all over the news and in the newspapers. There was no Twitter or anything back then, so we had to read the news on the newspaper or see it on TV. Anyway, so this was a really cool way that I think we can also look at and see how the world came together to do this. Now you can look up any sequence for any protein you want on the internet. So when we learn about DNA, okay, so a lot of people, there's a mutation in the DNA. We assume DNA is what the problem is. Now, although yes, we're going to learn that the error is in DNA. DNA is used to make RNA, and then RNA leaves the nucleus and enters the cytoplasm, and a ribosome attaches and makes a protein. Now, if the DNA is not correct, then the RNA is not correct, then the protein is not correct. So the problem is the protein, because when you change, we'll learn this chapter that when you change even a single nucleotide within our DNA sequence, we can get a completely different protein because the amino acids that create the protein have different charges and different characteristics that drive how this protein is going to fold. So, right, so here is a protein that's in its complex three-dimensional structure. So if I were to change one thing within this could fold a completely different way, and then that's a mutated protein. And that's how we get disorders. It's not so much the DNA is causing the problem. Yes, that's where the error is, but the protein that is created is not going to be correct. It's not going to function properly because it's not folded properly. And so then we have a problem. So this week we'll also be going through cloning, transgenic animals, GMOs, and other things that we've moved past. So we talked about earlier, right, where the Human Genome Project started in the 90s. And here we are now, and we've come so far. We can insert genes into plants. We can insert genes into viruses to treat diseases. So we're gonna be going through all of that over the course of this module. So some main things that you're gonna want to really focus on is take the time to really watch the lecture video. Understand that DNA can't leave the nucleus, right? So DNA has to be made into RNA, and RNA leaves and goes to the cytoplasm, and then that creates the protein. Okay, so make sure you understand that process. And then moving forward, understanding how we can then insert RNA into a virus to create some sort of treatment. And these are very complex subjects. So outside of the materials I've given you, go online and look up videos and see what can help you to understand better what I'm talking about when I say transgenics and when I say cloning and those types of things we're going to be talking about this week.